Okay, so what is the difference between absorption and assimilation? So this is all part of being a holozoic feeder. We do ingestion, digestion, absorption, assimilation, and egestion. The absorption bit then is the movement of those digested food molecules from the ileum, the small intestine, into the blood capillaries of the lacteal. It will then be transported around the body in the blood, and then assimilation occurs. So this is the movement of those molecules into the cells, and then they will either use it or store it. Can you explain how an inactive form of an enzyme becomes activated and why the active form could digest your cells? So pepsin and trypsin are the two peptidases that you need to know about. So they both digest proteins when they are active. And we are made up of a lot of protein. So if we stored them in the active form, either inside a cell or inside an organ, then what would happen is that they would start to digest the proteins of those cells or those organs. So that would be called autolysis, kind of self-digestion. So therefore, they're stored in their inactive form, and they're only activated when there is ingested protein from what you've eaten ready to be hydrolyzed. So pepsinogen is the one in the stomach. It's stored in that inactive form, and then when it comes into contact with hydrochloric acid, it is activated into pepsin. And trypsinogen is the inactive form in the pancreas, and then once it comes into contact with enterokinase in the duodenum, it's activated into trypsin. In ruminants then, do the bacteria inside them break down the cellulose, or do they secrete the enzyme which breaks it down for them? So the key thing about ruminants or any animal that wants to break down cellulose is it's got to have this cellulose digesting bacteria inside its gut structure somewhere. So the cellulose digesting bacteria secrete the cellulase enzyme into the area of gut that they inhabit. So they do a process of extracellular digestion. So they're secreting the enzyme out of the bacteria. These animals break down the cellulose consumed. These enzymes break down the cellulose consumed by the animal. And then the products of that can be absorbed by the organism. There are some weird examples. So rabbits have an odd gut structure, which means that they've got some behavioural evolutionary traits to overcome that. So if we have a look at the structure of a rabbit, and this was in one of the homework questions, you will see that these cellulose digesting bacteria aren't found until the cecum here. So they eat the grass, it moves through here, moves through here. So it's going through all of the small intestine where absorption usually goes on. And then it pops into the cecum. The cellulose digesting bacteria break down the cellulose, but unfortunately by this point, there isn't anywhere now to absorb it because the absorption part was here. So what a rabbit does is it releases these first pellets out here of its anus, and then it eats those ones. So it re-ingests those. They go back in to the stomach here. And then as they come through here, it means now that we can do absorption of all those nice products of that cellulose digestion. Right, next one. I've seen different roles of the different types of teeth on various mark schemes. Some say this, some say that. What are the correct examples that we should be using in the exam? So I would say that this is a pretty definitive guide to what you should be doing. So with the herbivore here, just remembering that it doesn't have any upper incisors here. It's got this horny pad. And then we've got the lower incisors. So these two work together here to cut plant material. And then you've got the lower canine and then the space here, the diasterna. And that helps with kind of moving the food around, manipulating it around the mouth. And then at the back here, we've got the premolars and the molars here, and they help to grind down that plant material. And then we've got the carnivore teeth here, remembering that they only eat meat. So now you've got the front ones here, the incisors, and therefore kind of pulling the flesh from the bones. You've got the canines here, and they're important because a dog is a bit of a bad example, really, because they get given their dog food. But if you were looking at a wolf that had to catch its prey, then they've got to grab hold of their prey. And often they will clamp down with these teeth and constrict the airway, and that's the way that they kill their prey. But they need to hold on to it whilst it tries to run away from them. Then we've got the premolars here, and they can slice the flesh. We've got the carnassials here, and they can shear the flesh off the bone because they're nice and big and sharp. And then at the back here again, we've got the molars, and they can be used to crush bone. Right, how is hydrolysis reaction different from any sort of catabolic reaction? The fact of the matter is you can think of them as exactly the same thing, really. So any sort of catabolic reaction is the breaking apart of molecules into smaller molecules to release energy, breaking down a protein into amino acids, or a triglyceride into fatty acids and glycerol, or a disaccharide into monosaccharides. These types of catabolic reactions are all examples of hydrolysis. The next one is trying to work out how you do this microscope question on page 126. So different teachers do this in different ways. I like to do it by, first of all, working out what my two scales are. So this one here, this big one here, is my stage micrometer, and the little one that is underneath it, that is my eyepiece graticule. Now, this one here, the eyepiece graticule, that is reading on here between 1 and 10. Now, really, they should read between 1 and 100. So if it's saying between 0 and 10, you know that whatever you read off that answer, you're going to have to times your answer by 10, and that will give you your eyepiece scale. And then if we look at the stage micrometer, then you can see on here, it's going up in tens. Now, what we need to do is work out how many of these are worth how many of these. So you need to look at your diagram and find a point where they match up perfectly. Now, I would say that point is here, where we've got 40, and down here, we've got 9. So we're going to say that the eyepiece graticule scale is 9, but because we know we've got to times it by 10, it's actually going to be 90. So we're saying that 9 eyepiece units is equal to what in terms of stage micrometer units? Now, in terms of stage micrometer units, it's going from 20 up to 40. So that is 20 stage micrometer units that we have got fitting into those 90 eyepiece units. So 20 stage micrometer units. And it tells us in the question that 1 stage micrometer unit is worth 10 micrometers. So now to convert that into those micrometers, we need to times our answer again here by 10. So that will give us 200. So now what we know is that 90 eyepiece units is worth 200 micrometers. So we need to work out what 1 eyepiece unit is worth. And therefore, to do that, we're just going to do, as it says here, the 200 divided by 90. And therefore, your answer when you do that is 2.2 micrometers. Right, the next one. What is bile? Where is it made? And what does it do? So bile is an alkaline, green, kind of brown fluid. It's made in the liver. So it's made up here in the liver, and it is then stored here in the gallbladder. It's not used here. So to get it to where it needs to be, down here in the duodenum, it comes down here and it goes via the common bile duct down into the duodenum. Its main function is to emulsify, and you must use that term in the exam, lipids from large droplets into smaller droplets. Now, the key thing there is it's not hydrolyzing the lipid molecules. So it's not breaking any bonds within the lipid. All it is doing is just converting it into large to small droplets. And often the analogy is like when you're using washing up liquid on a fatty pan when you're doing the washing up. You see the droplets of fat getting smaller when you use the washing up liquid. What it does do is it increases the surface area for then the lipid digesting enzyme lipase to work on. Its secondary function is it helps to neutralize the stomach acid as that pyloric sphincter of the stomach opens and allows the contents of the stomach to come into the duodenum here. Next one, what's the difference between excretion and egestion? I would say that excretion is more of an umbrella phrase about removal of any sort of waste from the body. So it could be urine, it could be feces, whereas egestion really is talking about the excretion of the feces. So egestion is kind of more specific to poo, whereas excretion could be urine, feces, sweat, etc., etc. Next one, how does the hydra digest its food? So this is our little hydra here. You will just be studying it or just have studied it as well in nerves. It's quite a simple organism, but it has these tentacles coming off here, which have special cells that can sting its prey. And then it has this opening at the top here. It only has one opening into its body cavity, the mouth region, and the food gets ingested in there, but the waste also gets egested out of it. So the prey is ingested via this mouth cavity. So it gets stung and kind of moved into here. And because it's being ingested into the body, then we should know that it's a holozoic feeder. Specialized cells then in this region here, they will secrete our enzymes into this hollow body cavity. So it's a type of extracellular digestion. And then the prey that's gone in here will become digested into those soluble food molecules, and the digested food will be absorbed and then sort of assimilated by the cells, used by the cells, and then any waste gets egested via the mouth back out here.